Back to another educational segment of You've Got Potential. We thought this would be a good opportunity to talk about a little bit something more on the macro scale. So prions or subviral particles. So why don't we talk about viruses? And I think the most relevant and best way to introduce viruses is something you guys have heard of, COVID-19 obviously. It's very interesting and it covers a lot of different information within viruses in the viral life cycle. So let's get into it. This is actually on this first slide here. You see on this image on the right, this is what's called a transmission electron microscopic image, and it uses transmission electron microscopy, which is you beam electrons through a focusing lens, and then there's a fluorescent backing behind the object, which can either have these electrons refract or it absorbs, and that'll create a really high resolution image. This is like revolutionary, and TEM's being used a lot more lately, and so it's definitely something to look into if you guys are interested. That's sort of just a side note. This is from the CDC. This shows the coronavirus within a cell sample. These little blue spheres here are represented, are supposed to represent the coronavirus, and so this is really cool. So first, before we talk about viruses, I want to get into like what defines a living being, what is life. I don't want you guys to contemplate this too much. This isn't like an existential philosophical question, but the point is that all living things, they have some sort of order, hierarchy, and structure. So even your bacteria are basically micelles with a cell wall and DNA. They can respond to their environment, so if there's a change in the environment, they try to maintain homeostasis, and internal conditions will change to reflect that. They reproduce on their own, meaning that they have their own machinery to undergo mitosis or whatever their preferred method of reproduction is, such as sexual reproduction. And they have growth and development, so they can replicate their organs, they can change them, they can not organs, organelles if they have membrane-bound organelles. And so if you're familiar with life cycles at all, that should make sense. They can maintain homeostasis, meaning that there's certain boundary conditions within the cell or cells, and they want to maintain those conditions in order to function optimally, right? So that goes into a lot about like optimal pHs for enzymes and heat and things like that. And they have some sort of metabolism or energy processing, right? So if you guys ever learn about glycolysis, or mitochondria or anything like that, they have some sort of mechanism in order to basically leverage chemistry in order to produce work and maintain homeostasis. And so all these are very interrelated, and it's really good to think about because these introduce some really cool principles on the broader scale. So getting into viruses, are they living beings? Unequivocally, they are not. Which characteristic of life do they not have? What do you think? Well, it's reproduction. So they hijack the host cell in order to reproduce and produce more viruses, and so that's why they're considered not living beings. But they do have a life cycle. Basically, it's this sort of process they go to in order to replicate. They're not living, so I guess they don't have a life cycle. But this is like some like very cool like artist rendition of the SARS-CoV virus. You have this like glycoprotein on the outside. You have a membrane, obviously, to encapsulate everything. They have the SARS-CoV virus is RNA, and so it has what's called positive sense RNA. We'll get into that. And then, so there are two general life cycles of viruses. This is not reflective of COVID. This is for bacteriophages only. But it's a good way to understand sort of how viruses operate. Generally speaking, you have lytic and lysogenic. So as shown in this rendition, what you have is that the viruses plop on top of the bacteria or bacterium and injects its genome, whether it's DNA, RNA, or both. This genome sorts of floats around, and so in the lytic cycle, what it'll do is it'll get into the nucleus, but they have enzymes that'll degrade the host genome and then replace the host genome with their own, and it'll hijack the host translation machinery in order to transcribe and then translate viral proteins that can be assembled and eventually produce viruses. And the rate of which this can happen can be so fast that the cell literally bursts from the internal pressure caused by viruses exiting the cell. Lysogenic is a little less intense, although it eventually will lead to the lytic cycle. And it's when instead of destroying the host genome, the RNA or DNA or RNA-DNA complex from the original virus, it'll actually integrate itself into the host genome, and it'll still get translated to some degree, but not as much. And the host can replicate, and this is very interesting, because what you can do is that if you can do that, you can have a whole population of bacteria that are like infected with this like viral genome, and then eventually environmental conditions can trigger the virus to go into the lytic cycle, in which what will happen is the degradation will occur of the host genome, and then you have the rest of the lytic cycle that'll go and eventually destroy the cells. So that's a good sort of starting/segue into understanding with viruses. I first want to go over this term positive and negative sense RNA. This is really important. It's basically general terms that describe, like, can this RNA be directly used by the host or can it not? And so what viruses do, they're either positive or negative sense. Their genomes are positive and negative sense RNA. And so when they inject their RNA genome into the host cell, positive sense says you can immediately start transcribing and translating this viral genome into useful proteins and useful mRNA, and the whole cycle can just start. And it's not lytic or lysogenic. This protein production is all occurring in the cytosol, and so what will happen is that this translation assembly will occur on the cytosol, and viruses will propagate. The COVID virus will propagate. So the way that the transcription has occurred, or the replication of this positive or negative sense RNA is done, is through this protein complex, which is depicted in two different ways here. So here, this is this is the helical, I guess, rendition of it, and this is a different, more dotted rendition of it. This is from the RCSB protein data bank, which is, you guys should look into it. It's really cool. And negative sense basically describes when the RNA has to be turned into positive sense RNA first, and then translated. So this is called RNA-dependent RNA polymerase, right? And this protein does it all. So it comes with the virus, and so it'll either, if it's positive sense, it'll start transcribing more mRNA, and if it's negative sense, then this same polymerase will turn negative sense RNA into positive sense RNA, and then translation can occur by using host machinery. And again, I highly encourage you guys to look at these structures, because they're fascinating, and there's a lot of good information on the RCSB website. So, products of translation. So you get the RNA, let's say it's positive sense, you can immediately start using it. What are your products? This is how many products there are. So maybe you guys have taken a bio class and you've learned the structure of a virus, and it seems so simple. You might have a capsid, you got your RNA, simple RNA in there, but there are a lot more products than you think. So for this is the SARS genome and proteome, and this is basically showing like what parts of it encode for what and what proteins, and you can see that there's plenty of them. And then so right here where my cursor is, that's your RNA-dependent RNA polymerase. These little IDs, those are the IDs that are uniquely defined them in the RCSB database. So if you ever want to look any of these up, you're like interested in them, go for it, look it up. It's right in there. And then so let's go into the life cycle. That'll be sort of where we wrap it up. You guys have a sufficient background now, so let's get into it. It's really intuitive to some sense based on the information we've presented here. So you have this SARS coronavirus that binds to some receptor on the cell surface, and basically you have an endocytosis thing going on where the cell envelops it, brings it in. It's kind of crazy, right? It's like inviting the wolf in, and so then if it's positive sense, like we said, host machinery can start translating it. You can also have RNA-dependent RNA polymerase start making more of that viral RNA, and that's very useful, because the more RNA you have floating around, viral RNA, the more product can be translated in terms of quantity. More proteins can be translated in terms of quantity, and so you'll have even more viruses. So you don't want just like one strand floating around. You want many, many strands floating around. If you're the coronavirus, if you're people, you don't want that. So you have this translation. If it's negative sense, you have this modification of it into positive sense and translation. Eventually these proteins sort of assemble on their own, and then they're released via exocytosis. Antibodies are presented to the immune system, and this releases a cascade of events that lead to your immune response. And so that's how your body starts to try to figure out, like, okay, what is in me? How do I get it out? What do I got to do? There's a whole process on it, and we're not getting into it. But I hope you guys enjoyed this presentation, that you learned a little something. This presentation, you can go through here and you can see all these links. There's plenty of background information to find and look at. The papers are all cited in here. So have a good day. I hope you guys learned something, and remember that you've got potential.